Thank you, Jean, for that introduction. So as Jean has said, we have worked together before, and we continue to work together. Jean is collaborating with us on one of the, which is a kind of a sequel to one of the projects that I'll be talking about today. And her knowledge of nutrition has just been a real boon to the project. Not just a boon, but I mean, it is just a valuable piece of the project. And we're so fortunate to have her and to share her with some larger collaborations that are really looking to her to answer some of their questions about nutrition and asking the right questions. And so, yeah, I just wanted to say that about her. She's kind of humble too. So I guess first I want to acknowledge the research staff that has been extremely instrumental in conceiving, conducting, and analyzing this work. No disclosures. So food allergy can result in life-threatening reactions, and it adversely affects the quality of life for patients and their families. And the rise in food allergy has been well publicized. We have seen an almost 40% increase from 2007 through 2018. The highest increase has been among 5 to 17-year-olds, where we've seen a 45% increase during this time frame. Also, the literature suggests that children are taking longer to outgrow their milk and egg allergies, which are some of the more prevalent allergies in infancy and childhood. So food allergy falls in the continuum of allergic conditions, often referred to as the allergic march. The allergic march is a depiction of the natural history of allergic manifestations culminating in atopic asthma. This little graphic kind of depicts the transition and change and process. And if we look at this progression, and I'm using the term very loosely, it's just difficult not to imagine that if we can stop this progression or prevent this progression, we can prevent allergic asthma. And that's actually how I got interested in food allergy. I was initially working on asthma. So first, in thinking about the study of food allergy, it's important to define relative concepts. So adverse food reactions are any aberrant reaction following the ingestion of food. Food intolerance is actually a non-allergic food hypersensitivity caused by some unique physiologic characteristic of the host, a metabolic disorder, lactase deficiency. Food sensitization is a production of food-specific immunoglobulin E antibodies, or IgE, and they initiate this type 1 food hypersensitivity reaction. Food allergy is defined as an adverse health reaction, adverse health effect arising from a specific immune response. So there's activation of mast cells and basophils stimulated by IgE that occurs reproducibly upon exposure to a given food. So food allergy actually requires both the presence of sensitization and the development of specific signs and symptoms upon exposure to that food. So sensitization alone is not sufficient to define food allergy. And this kind of presents some challenges for epidemiologic studies of food allergy. As we were saying, food sensitization is not the equivalent of food allergy, although it's a much easier endpoint in conducting research. It's been estimated that 40% of persons sensitized will respond to an oral food challenge. So an oral food challenge, this next bullet, is a double-blind, placebo-controlled food challenge. It's still considered the gold standard for diagnosis of food allergy. It requires trained medical personnel, and it can take up to 4 hours of a patient's time. And there's also safety concerns because anaphylaxis is possible and has occurred during an oral food challenge. So this is pretty challenging for large epidemiologic studies in terms of feasibility and safety and cost. Ascertainment is also a challenge. So questionnaires and surveys can be used, but respondents can confuse food intolerance with food allergy. The incidence and prevalence is relatively low. Incidence could be less than 1% depending on the population. Prevalence estimates are much easier to obtain, but could be at the most right now could be 8 to 10%. And because food allergy is usually diagnosed during infancy, it's optimally studied in birth cohorts. And we know that birth cohorts are a pretty labor-intensive and expensive study design, but as we have found, they're so worth it. So we currently have several birth cohorts at Henry Ford Health System. The one I'm going to focus on today is the WHEALS birth cohort, which stands for Wayne County Health and Environment Allergy and Asthma Longitudinal Study. The wheal is that elevation or lump on the skin surface that you get as a result of a skin prick test to determine whether or not you're allergic to something. It's a long-term study that started in 2003 and recruited pregnant mothers from selected OB clinics in urban and suburban Detroit. It's an unselected cohort, so it's not just focused on high-risk kids. Therefore, our results are a little more generalizable, and it's racially and socioeconomically diverse. The data collection for WHEALS consisted of interviews with mothers prenatally and after the infant was born, as well as home visits where blood, breast milk was collected. We also collected dust from the home, and we collected infant stool samples. So here's one of the very early studies that we did with WHEALS, with the WHEALS cohort in relation to food allergy. So in thinking about that allergic march and risk factors for allergic asthma, we were interested in examining factors related to food sensitization, since food sensitization is a prominent risk factor for later development of allergic asthma. And the idea was that early antigen exposure might induce aberrant immune responses leading to food sensitization. So we wondered if exposure to food antigens during a crucial period of immune development was a risk factor. And we were also thinking that maybe it might contribute to racial disparities that we see, and that perhaps African-American moms are more likely to introduce foods complementary to breast milk at earlier ages. And at the time we were doing this, there were surprisingly few papers on racial differences in early feeding. Some of these papers suggested a difference, but it just wasn't consistent. So here's the adjusted odds ratio, and these are the results of this study. What we found was that there wasn't a difference. We didn't find a racial difference in the timing of, or the timing of the introduction of complementary foods. It was higher in African Americans, but it wasn't significantly higher. What we did find was that the association of early introduction of complementary foods was related to a reduced risk of food sensitization. But this association was only seen in infants whose parents reported a parental history of atopy. So this was not the outcome that we were expecting. We were thinking that early introduction would be related to higher risk. But what followed our study and supported our results was a study that got a lot of media attention, the LEAP trial, in which they randomly assigned infants to either consume or avoid peanuts until age 60 months. And what they found was that the avoidance group, actually, they were more likely to develop a peanut allergy. So evidence was pointing to, this crucial time period of gut immune development in which exposure to certain food antigens could be of benefit. And we think of dietary antigens as, we can think of dietary antigens as being foreign to the immune system. It's one of the earliest exposures experienced by the gut. Yet we can tolerate food antigens, and our immune system is non-responsive if we don't have food allergy. So this process is not completely understood. And this little graphic depicts events that are involved in reaching the state of immune non-responsiveness to food antigens. So food, here's my little antigen, my food being introduced into the lumen of the gastrointestinal tract. And there are, these antigens can be absorbed or transported through these intestinal epithelial cells in different ways. Depicted on this slide, there can be paracellular leaks. There can be endocytosis or transcytosis based on these M cells or these goblet cells. And then also dendritic cells can sample antigen in the lumen. And these activities actually enable antigenic presentation to underlying immune cells. And what happens is, if you are going to reach a state of non-response, this presentation of these antigen start a process in which naive T cells are differentiated into T regulatory cells, and they secrete immunosuppressive cytokines like IL-10 and TGF-beta. And so this process of immune non-responsiveness is referred to as oral tolerance. And so food allergy is failure to achieve that state of oral tolerance. So this got us thinking about some other potential uses of our data. And you remember that the first study I talked about used food sensitization as an outcome. And food sensitization is not the same thing as food allergy. So we really wanted to tighten up our endpoint a little bit. So what we did was we collected all of the information that we had in the cohort. So parental surveys told us about symptoms and reactions to food ingestion, and of course we had information from a clinical visit at 2 years where we got blood and looked at IgE, and we also looked at skin prick test results. And we abstracted data on our cohort participants from the medical record. So we put together this package and gave it to board certified allergists and asked them to make a determination as to whether the infant was highly likely, or unlikely to have food allergy. And so we were able to use this outcome in the next study. So this next study was based on parameters involved in oral tolerance. The literature has not had consensus on breast milk as a risk factor for food allergy. There are some studies that show it is a risk factor, and there are some studies that show that it is protective. Our hypothesis was that perhaps certain components of breast milk will vary from individual to individual. And specifically, we were interested in TGF-beta, which is an important cytokine, as I mentioned, that also works with IL-10, which was also mentioned. And these cytokines stimulate processes that lead to oral tolerance. And we thought maybe levels of TGF-beta differ in breast milk, and perhaps this was associated with infant allergic outcomes. So we were able to measure levels of TGF-beta in breast milk of mothers that were enrolled in WHEALS. And although our cell sizes started to get really small, because we had to have, we only included those women that had breast milk where TGF-beta could be measured, our results were in the hypothesized direction, in that lower levels of TGF-beta were associated with atopic manifestations in their infants. This is true for sensitization, for skin prick test, and we got the strongest suggestion of an effect with the outcome that we developed through our physician panel. So this was only found, though, among white mothers who were non-atopic. So these results kind of resulted in more questions, but they seem to be supporting the way the literature and our team was thinking. So just if we think about this path to oral tolerance, we know that genetics plays a role because we know that asthma and allergy tend to run in families. But these environmental factors also influence this pathway. And the gut microbiome is key to achieving gut immunity. And there's really this feedback loop where they are regulating each other. So just kind of returning to this slide, we had talked about how IL-10 and TGF-beta are important contributors to oral tolerance and how they play a role in stimulating immune suppression processes. But another important component of breast milk is IgA. So I like this slide, but it might be very difficult for you to see this. But IgA is another immunoglobulin that seems to work in concert with IL-10 and TGF-beta. So that, we're starting to see this connection, TGF-beta and IL-10 in breast milk actually promote class switching to IgA. Excuse me. And IgA promotes intestinal barrier function and also has seems to play a very prominent role in homeostasis for the host in terms of commensal bacteria. And so this gut microbiota, the gut microbiota also support generation of T regulatory cells, which of course secrete the TGF-beta and IL-10 and other inhibitors that lead to oral tolerance. So all of this ties into hypotheses that have been presented that try to address the increases that we've seen in allergic conditions, including food allergy. And central to these hypotheses surrounding the development of allergic disease is this hygiene hypothesis. And this hypothesis has undergone several modifications, largely based on refining and redefining what the underlying concepts contributing to an increase in immunologic disorders are. And this reexamination and reevaluation has led to a general consensus on a theory of microbial dysbiosis. So given that microbial colonization of the infant gut is known to play a key role in immunologic and metabolic pathways, the thought is that disruptions during this complex process, such as alterations in the compositional development of the microbiome during infancy, could predispose infants to diseases later in childhood. So that a dysbiotic gut microbiota is thought to lead to immune dysregulation and atopic disease. So it was collection of those stool samples in the WHEALS cohort that led to the establishment and funding of a new study, Microbes, Allergy, Asthma, and Pets, that was funded. And I have to say that in 2003, people were not really thinking about the microbiome as a contributor to allergic conditions, especially food allergy. And so the principal investigator, Christine Cole-Johnson, I mean, she really had a vision here, in deciding in 2003 that they were going to collect stool samples and freeze them. So it just turned out to be invaluable and a real contribution to the field, I think. So MAP is actually a sample, an analytic sample that is drawn from WHEALS. It uses the infant stool samples that were collected in WHEALS at 1 and 6 month home visits. The sample is representative of WHEALS in a lot of the demographic and baseline characteristics. There was one exception in that the MAP kids tended to have higher incomes, their families. We used 16S rRNA gene sequencing to identify bacterial composition in the infant gut. We had about 300 stool samples at this point for analysis. So now I just want to talk a little bit about some of the findings that we had with using the stool samples and the microbiome. So not all of them are related to food allergy, but I just thought they would be interesting. So we looked at 1 month stool composition by mode of delivery, so C-section and vaginal. And the literature, there had been a good deal of literature coming out that suggested C-section, babies born by C-section had a higher risk of developing allergies as opposed to babies born vaginally. So this is a result showing how 1 month stool composition differed by mode of delivery. And you can see that this Bacteroideaceae was certainly more abundant than for vaginal birth babies than for C-section babies. So this is a multi-dimensional plot that basically tells us how similar the infants are in terms of their gut bacterial composition. So the pink dots represent breastfed infants, infants who were still breastfeeding at 1 month. And the green dots represent infants that were not being breastfed at 1 month. So the more similar these subgroups are, the closer they will be to each other on the plot. And so this plot just shows nicely how the composition of the gut flora differs by breastfeeding status. So this next analysis also looked at TGF-beta, but was looking at the relationship of this cytokine to gut microbial composition. So the bars here are color-coded. So negative is red, a negative association is red, which means there's less of a particular bacteria. And then the blue bars are related to a relative abundance of a certain bacteria. And so you can see with the, now I'm going to stumble over these names, Streptococciae and Ruminococciae seem to be higher in abundance, while Staphylococciae seem to be in lower abundance. So it's really interesting given the ties to TGF-beta in breast milk and to IgA and the microbiome. So here's a study that we, an analysis that we conducted, and we have written this up and are looking for a home for this paper. This looks at the infant gut microbiome and IgE-mediated food allergy as determined by our physician panel. We were able to, by the time that we were doing this study, we were able to increase the number of stool samples with microbiome information. So this first figure shows bacterial diversity by food allergy status. This is alpha diversity, which is really about what species are present and how many different types of species are present. And these are trajectories of bacterial richness over time for egg, milk, and peanut. So children with food allergy, which is the blue line, for all of these allergens, I'm sorry, the blue line is no food allergy, the red line is food allergy. So for all of these allergens, kids with IgE-mediated food allergy seem to be deficient in diversity compared to those with food allergy. And in the same analysis, we looked at individual OTUs or operational taxonomy units. Each of these clusters here is intended to represent a taxonomic unit of a bacterial species. They're categorized, you can't read this, but they are categorized by color. And here are the results at 1 and at 6 months. So the blue circles represent an overabundance of certain OTUs, and the red dots represent deficiency. So and then the effect size is represented by the size of the dot. So as you can see, there were deficiencies for children, for certain bacteria with food allergy. There were deficiencies that were associated with food allergy at 1 month and at 6 months, and it seemed to be particularly at 6 months. Another analysis that we did that was really very exploratory was to look at predicted metabolic pathways. These KEGG pathways represent our knowledge of metabolic pathways that are associated with certain compositions of gut bacteria. This also is color-coded. So here, the blue represents an overabundance of microbiota representing a particular pathway. And the red represents a lower abundance or a deficiency of those microbiota. And we could see that for children with food allergy at 1 month, there actually is some peak here, but I see that it didn't show up. But certainly at 6 months, you can see for children with food allergy, there are some definite deficiencies. And so this suggests that there could be some differences in the predicted metabolic potential of food allergy compared to no food allergy. So we did find variations in the gut microbiome of infants between IgE-mediated food allergy and no food allergy. The alpha diversity trajectories were significantly lower. We saw deficiencies in the abundance of some of the OTUs, and our exploratory analysis suggested that the metabolic potential for gut microbiota of infants with food allergy is altered. So in terms of future directions, recent advances in food allergy now really place the microbiome. It's not just food allergy, other diseases too, but they place the microbiome at center stage of pathogenesis. And it's important for the field to better define dysbiosis that is associated with disease. So in these studies, I will say that studies have found different OTUs that have been associated with an atopic condition. But what the experts are saying now is it's really about the function of these groups of biota, so microbiota, so that there needs to be certain functions that are represented in the gut microbiome. It's not necessarily a specific species, but it's the function that those species may represent. And different species may actually have a similar function. So make sure I went over, yeah, we don't know if dysbiosis can differ by specific allergen. We focused on IgE mediated food allergy, but the risk may be different for non-IgE mediated food allergy. And given the importance of IgA in the host commensal relationship, if we're talking about the microbiome, more research is really needed in this area. So in terms of more research, we are starting new birth cohorts at Henry Ford Health System. And it's just kind of funny how these birth cohorts are all taking place at the same time. So you can imagine that this is taking a lot of thought in terms of logistics. The REACH birth cohort, the one that Jean is very heavily involved in. Well, actually, Jean, you're heavily involved in all three of them. But the REACH birth cohort is going to be an unselected cohort. It's not going to be focused on high-risk infants. And it is more focused on maternal factors that influence the gut microbiome of the infant. And we're thinking that with this cohort, we should also be able to do some sub-studies on kids that are diagnosed with food allergy. We'll do some ancillary studies. The STAR is a high-risk cohort. It's actually going to be a subset of people that are in REACH. These are going to be moms that are found to have a history of allergy and have elevated IgE. It's also focused on maternal factors, and people in STAR will be asked to give us additional samples. So, we're collecting blood and stool like we did for the other cohorts, but in STAR, we are going to collect them more frequently. And then Sunbeam is a new cohort that is starting, and this one is specifically focused on food allergy. It is going to be an unselected cohort focused on eczema and food allergy. And food allergy in this cohort is going to be determined by oral food challenge. So that's going to be a real benefit to this cohort. And this cohort, Sunbeam, is not just at Henry Ford. So there are, I cannot remember how many other, maybe 10 other organizations that are contributing to this cohort. And I think that's all I have for right now. I think we have time for questions. No?
Yes, thank you so much. That was a wonderful, wonderful presentation. I don't know if you can see, if you want to stop your screen share so you can see everybody's face. That's up to you. Maybe you don't want to see that. And then whoever has a question, go ahead and just turn on your video and unmute yourself. And while people are coming up with questions, I'll let you know that one of our participants, Dr. Diane Haggerty, put in the chat about how great your acronyms are.
I can't take credit for those acronyms. I can tell you that a lot of feedback, a lot of people had input into them.
Certainly is an art to that. And they
Yeah, for sure.
Questions?
Hi, Dr. Joseph. My name is Chen Fei Ma. Question about, I wonder if you investigate the formula, the different nutrition in the formula can contribute to the difference in the microbiota community.
That's a great question. We actually, at one point, we were going to try to get a sample of formula from each mom that participates. So right now we are really thinking about what samples, what biospecimens we're going to collect. And so, you know, just kind of making a decision based on participant burden. So I'm not sure if that's going to stay on the list, but it's definitely something that, you know, that we're thinking about, looking at the composition of infant formula. Thank you.
Thank you.
Hi, thanks for that great talk, Dr. Joseph. My name is Sarah Comstock. I'm an assistant professor here at MSU, and I was so excited when the WHEALS study came out. Gosh, a long time ago, but I was working in the area of food allergy, and when I saw that study, I was like, yes, this was exactly the study we needed. So that was super exciting. And yes, your colleague was very forward-looking when she thought to collect those fecal samples.
You know?
So important. For your new studies, I was wondering, there's been a lot of focus on dietary diversity early in an infant's life and how that might be protective from food allergy. And so I was wondering, with some of your new cohorts, what type of diet data you're planning to collect and how you're planning to factor that into your results you're going to get from those studies.
Well, that's a great question, because we have a well-known expert that is guiding us in that aspect of data collection for our cohort. So we are collecting diet information at several time points. I think we're actually collecting it prenatally too, aren't we, Jean? Yeah, and post-delivery as well. And so you can get details about that from Dr. Jean Kerver. Yeah, we're finding that that's so important now. If you're looking at the microbiome, it's just so important. And we're, you know, we really want to know what maternal factors influence infant gut microbiome. So thank you. Hi, Nigel.
Nigel, did you have a question?
Good to see you.
Good to see you too.
No question, Nigel?
Not at the moment. I have no question.
That's fine. It's just surprising.
Christine answered everything.
as she does.
Case.
Yes, and so Christine, I will just say, Dr. Comstock, who asked that previous question, is in our Department of Food Science and Human Nutrition at MSU, and she and I are closely together, and I hope that she can be involved as a collaborator on some of the work coming out of REACH and STAR, because she has given me an education on all of the various aspects of microbiome work that I wouldn't know anything about without her.
That's great. Yeah, we welcome collaboration and, expertise in these areas in which we are lacking. So this has got to be a collaborative effort. And, the nice things about, as you well know, because you guys do observational studies and have birth cohorts as well. They're just so valuable for answering a myriad of questions. And while we're focused on asthma and allergy, we have others in our group that are, looking at other conditions and will be able to use this information. I think it was Gail that was saying that she was interested in obesity and breastfeeding. And I think that we have an investigator that is looking at that with the WHEALS data and maybe has already published on that. I'd have to check on that.
Yeah, and Dr. is one of Gail's mentors. She's on her mentoring team and was on her dissertation team. So we're all a big happy family. And
Great.
all working on Echo now as well.
Great.
Other questions? Gail, do you have a question?
No, not at this time, but thank you again for sharing your study. It was very insightful, or actually all of the work that you've done thus far. So thank you. It was a pleasure.
Thanks. And, since you have expertise in breastfeeding, I think it would be great to collaborate on some things.
It's nice to see you again. And we just talked last week because we have a mentee in common. I wanted to ask about the obesity in terms of the food sensitivity issues in asthma. And obesity wasn't looked at for a long time with regard to asthma. More recently, it's been an interest. And for a while, I kind of ignored it, thinking, well, it's just, the racial disparity, and they just, are over-interpreting things, and it's not really about the obesity. But it looks like more recently that there definitely seems to be some strong evidence that obesity in childhood relating to asthma. And I didn't know if you want to say a little bit about that, and if it's related to food sensitivity work at all.
So, yeah, they have been looking at that, but no, I can't really say very much about it. We actually did in Puff City, we actually did have some information on weight. This was a study that was in urban teenagers. We conducted a trial in Detroit public schools, high schools, and we were able to look at obesity and asthma morbidity. And we did find a significant association. It was only in boys. And, I remember in looking at the literature around that time, it's been some time, results were were a little inconsistent at that time, but I think it's remained a hypothesis that has gained some support. They're both inflammatory processes. So yeah, I think that people are still examining that association. I'm sorry.
No. And I think what you brought up is also really important, that it's a very messy thing with asthma, which is why I know you've done the birth cohort work, is that people conflate the exacerbation of asthma with the, incidence and causes of asthma, right? So
Right.
It looks like the obesity work is still a little inconsistent, but it's growing, but it's more towards exacerbations of your asthma condition. And I don't think the evidence is that compelling yet about it leading to a diagnosis of asthma, that it causes asthma to occur.
No, I've seen it both ways, and I'm going to suggest that you have Andrea Bushrow Cassidy come and talk, because that is one of her areas of expertise, and she has been using some data from our birth cohorts to look at
Tease it
those
apart.
questions. Yeah.
Okay, good.
Hi, could I ask a question?
Sure.
It's about your well, it's kind of based off of your exploratory metabolic potential. And I was wondering if you have any plans with, whatever samples you already have collected, or with the upcoming cohorts, if you guys have any plan of looking into, that actual like metabolic, composition of these infant fecal samples, since there are a lot of associations with certain like short chain fatty acids and other things related to asthma and allergy.
Yeah, that's a great question. And that is not my area of expertise. And I seem to think that we are working with somebody at MSU that is going to help us with our metagenomics and metabolic analyses. And so if you like, I'll find out who that is.
That's actually Todd, that's Todd Leidig, right? And
That's right, yeah.
Yeah, because we use him for something else, so that's why I know. That's okay. We were trying to get a grant out with him today, so that's why.
Great. So yeah, he's going to be working with us on that. Should be really interesting, and IgA is also related to these short chain fatty acids, and they're an integral part of that process, that pathway to achieving oral tolerance. So it's all kind of coming together.
Thank you. How do you spell his name? Todd
Leidig. I think it's L-E-D-D-I-C-T. Is that right, Dawn? No? Okay.
No, L-L-Y-D-I-C.
Oh.
He's in the College of
Wow.
Natural Sciences and in charge of the lipidomics core, but they've started doing metabolomics. He has a new method to add the lipidome to the metabolome. Add the metabolome piece to the lipidome.
Oh, thanks for that information,
Thank you
Dawn.
so much.
Very nice guy to work with too.
Oh, great. I like working with nice people.
I wanted to ask one quick question. When you were talking about secretory IgA, do you have any plans to use pull down methods to pull down the bacteria that might be associated with that secretory IgA and try to figure out if some of the bacteria that are associated with the secretory IgA maybe are having a different effect than those bacteria that are non-associated?
So you sound like you have some expertise in that area. I am trying to put a team together that would look more closely at developing research questions around IgA and use some of these samples that we're going to be collecting to do some studies on IgA. So maybe I can keep you abreast of that.
I'd love to hear more
Yeah,
about that.
I'm just kind of interested in that.
I think it's really important because if you're trying to think about what the immune response is being stimulated to and how the immune system is responding not only to diet, but also to those bacteria, to know if there's some protective mechanisms occurring in the GI tract is really important. And I'd love to talk to you more about that.
Yeah, and I will tell you that IgA, in terms of sensitization and diagnosing food allergy, it's always been IgE. And researchers have, tried to see if IgA could be another biomarker. But the results have been inconsistent, and usually I think the consensus now is that it's not a good biomarker. But knowing more about its relationship to the microbiome and to this whole process of oral tolerance, I think it's worth, taking a really good look at it in different ways and seeing how it fits into this puzzle.